Artificial Intelligence Doctor. User question. What are the molecular mechanisms by which insulin resistance develops in peripheral tissues during prediabetes? GPT-01 answer. Introduction. Insulin resistance is a core feature of prediabetes and is characterized by the diminished ability of peripheral tissues, such as skeletal muscle, adipose tissue, and the liver, to respond effectively to insulin. This leads to impaired glucose uptake and elevated blood glucose levels. The development of insulin resistance involves a complex interplay of molecular mechanisms, including defects in insulin signaling pathways, inflammation, lipid accumulation, and oxidative stress. 1. Impairment of the insulin signaling pathway. Insulin receptor, IR, dysfunction. Insulin initiates its effects by binding to the insulin receptor on cell surfaces. In insulin resistance, there may be a reduction in the number or affinity of these receptors, hindering signal initiation. Insulin receptor substrate, IRS, alterations. After insulin binds to its receptor, it activates IRS proteins. Serine phosphorylation of IRS, instead of normal tyrosine phosphorylation, impairs its ability to propagate insulin signaling, leading to decreased glucose uptake. PI3K ACT pathway disruption. The phosphoinositide 3 kinase, PI3K, and ACT, also known as protein kinase B, pathway is critical for mediating insulin's metabolic actions. Defects or reduced activity in this pathway diminish the translocation of glucose transporter type 4, GLUT4, to the cell membrane, decreasing glucose uptake into cells. 2. Inflammation and cytokine production. Chronic low-grade inflammation, adipose tissue, especially in obesity, secretes pro-inflammatory cytokines like tumor necrosis factor alpha, TNF-alpha, and interleukin-6, IL-6. Activation of stress kinases. These cytokines activate serine kinases such as C-Jun N-terminal kinase, JNK, and IB kinase beta, IK, which phosphorylate IRS proteins on serine residues, inhibiting insulin signaling. 3. Lipotoxicity and lipid accumulation. Elevated free fatty acids, FFAs, high levels of FFAs in the blood, often due to increased lipolysis in adipose tissue, lead to ectopic fat deposition in liver and muscle tissues. Intracellular lipid metabolites. Accumulation of diacylglycerol, DAG, and ceramides activates novel protein kinase C, NPKC, isoforms, which phosphorylate IRS proteins on serine residues, impairing insulin signaling. 4. Adipokine dysregulation. Decreased adiponectin levels. Adiponectin enhances insulin sensitivity. In prediabetes, adiponectin levels are often reduced, contributing to insulin resistance. Increased leptin and resistin. Elevated levels of these adipokines can promote insulin resistance by interfering with insulin signaling pathways. 5. Oxidative stress and mitochondrial dysfunction. Reactive oxygen species, ROS, production. Overnutrition and excess FFAs increase mitochondrial ROS production, which can damage cellular components and interfere with insulin signaling. Antioxidant defense impairment. A reduced capacity to neutralize ROS exacerbates oxidative stress, further impairing insulin action. 6. Endoplasmic reticulum, stress. Protein misfolding. The accumulation of misfolded proteins in the triggers the unfolded protein response, UPR. Activation of stress pathways. UPR activation leads to the stimulation of JNK and IK pathways, contributing to insulin resistance through IRS serine phosphorylation. 7. Genetic and epigenetic factors. Genetic predisposition. Variants in genes involved in insulin signaling and glucose metabolism can increase susceptibility to insulin resistance. Epigenetic modifications. Environmental factors like diet and physical activity can lead to epigenetic changes, e.g., DNA methylation, that alter gene expression related to insulin sensitivity. 8. Gut microbiota alterations. Dysbiosis. An imbalance in gut microbiota composition can lead to increased intestinal permeability. Endotoxemia. Translocation of lipopolysaccharides, LPS, into the bloodstream promotes systemic inflammation, contributing to insulin resistance. 9. Hyperinsulinemia and insulin degradation. Compensatory insulin secretion. To overcome peripheral insulin resistance, pancreatic beta cells secrete more insulin, leading to hyperinsulinemia. Insulin clearance reduction. Impaired insulin degradation in the liver can exacerbate hyperinsulinemia, further downregulating insulin receptor activity. Conclusion. The development of insulin resistance in peripheral tissues during prediabetes is a multifactorial process involving hormonal imbalances, inflammatory signals, lipid metabolism disturbances, and genetic factors. These molecular mechanisms collectively disrupt insulin signaling pathways, leading to decreased glucose uptake and elevated blood glucose levels. Understanding these processes is crucial for developing targeted interventions to prevent or delay the progression to type 2 diabetes.